So the next drug class that we're going to discuss are the sulfonylureas and the non-sulfonylureas. So these are fairly old drugs, and they're named by their chemistry. They work very similar, so we're just going to consider them as one group, although chemically they're different. The ones are sulfonylureas and the others are non-sulfonylureas. And here are some names. So some famous sulfonylurea names are glipizide, glyburide. They all start with gly. And then for the non-sulfonylureas, nateclinide, rapaglinide, so they have also the glyinide in the name. And so what are they doing? So they just shut down this ATP-sensitive potassium channels. So here is how, let's use an example, glyburide works. And I have mentioned in the beginning of this video series that usually when glucose is sensed in the body, glucose is gonna be broken down and it's gonna generate ATP, which shuts down this potassium channel. And what they do, they just do it independent of glucose. They just shut down this channel. The consequence is that we're gonna have an accumulation of positive charges because now the potassium cannot leave the cell. And this is gonna trigger depolarization. The calcium is going to come in, and calcium triggers exocytosis. And here I've drawn some insulin in vesicles. And what's going to happen, these vesicles are going to fuse here with the plasma membrane, and then, so let's make them fuse here, and then the insulin is going to be released. So this really means that they, independently of glucose, trigger insulin release. So I've just written down here the mechanism of action again. So we inhibit the ATP-sensitive potassium channel, leading to depolarization, activation of the voltage-gated calcium channels, and insulin is secreted. And this is also called glucose independent. That's important. Insulin release.